Hello, good morning. You are welcome in the SOCT Zoology classes, episode number 131, Introducing Biochemistry. Biochemistry is the chemistry of living system. Whatever reactions take place in our body, they are included in biochemistry. But how to study the living reactions taking place in the living system? Actually, three techniques are used. In our living system, many molecules are there. And for studying any single molecule, we have to separate it from the mixture of all the molecules. So, three techniques are used in the biochemistry. First of all, is the centrifugation. First of all, the tissue is like the mixie used in our kitchen. There is a tube, and in it there is a vessel. It is revolved. So the tissue present in this area is made in the form of a solution, very fine, and after that it is centrifuged. Different fractions are separated, and there is a technique of subcellular fractionation also at different speeds, RPM or G. First of all, the mixture is centrifuged in a centrifuge tube, and the heavier portion is collected in the bottom, and the light portion is called supernatant. The precipitate is also called pellet, and the supernatant is again taken into. So first of all, the nuclear fraction is separated, then the remaining supernatant is again taken into another centrifuge tube and centrifuged at still higher speed. Mitochondrial fraction are separated, then further using ultracentrifuge, the microsomal fractions are separated and the cytosol remains as such. So different subcellular fractions can be separated and analyzed. Their chemical analysis can be analyzed. Second thing is that from a mixture of different molecules, the different molecules can be separated from each other. If they have a polarity, then they can be separated by electrophoresis. If they are non-polar and there is difference in molecular weight, they can be separated by on the basis of solvent drag. So there are two techniques. One, electrophoresis. Suppose there is we can take a paper or we can take a gel also, like the toothpaste. So the mixture of molecules is kept here, and its both ends are dipped in a trough having a solvent, and then two electrodes are connected with each chamber, connected with a electrical system. So what happens? This is a positively charged area, this is negative electrode. Those molecules which have a negative charge, they will migrate towards this end. Those which have a positive charge, they will migrate towards this end. So after some time, what will happen? The molecules will get separated and that can be removed from each other and analyzed chemically. Third option is the chromatography. So in chromatography there is a dispersed phase and there is a dispersion medium. There may be paper chromatography, thin layer chromatography, column chromatography. In paper chromatography what happens? Like the filter paper, there is very fine Wattmann's filter paper, and it has very fine pores, have a capillarity. It is dipped into a trough having solvent. There may be ascending chromatography or descending. Either from the trough, the paper movement of molecules may be in the descending order, and then may be ascending chromatography. So here also what happens? Suppose the mixture of compounds are kept here. The solvent, due to capillary force, it will migrate towards the upper end. And along with this solvent, some of the solute particles will also move towards the upper end. Those molecules which have a lighter weight, they will reach up to here. Those molecules which have a heavier weight, they will reach up to here. So till the solvent has reached at this stage, it is removed, and this is also cut. This is also cut and dipped into another test tube having the solvent. This process is called elution. Besides paper chromatography, like burette, a column may also be used, in which some Cefadex gel or silica gel are used, and a solvent containing the different molecules are poured here depending upon their molecular weight. The heavier molecules will migrate earlier, so each fraction is taken in a tube. Molecule having less weight will migrate slowly, so different fractions can be separated. It is called column chromatography. There is one technique, TLC, thin layer chromatography also, where a glass plate is kept, and like the lime coating of our wall, silica gel or some other medium is spread on it uniformly, and then it is dipped into a solvent. Solvent will migrate like this. Mixture of compounds are kept in the bottom. So after some time, same like paper chromatography, the lighter molecules will reach here, the heavier molecule will reach here, so they can be separated from each other. So these three are basic principles of biochemistry for separating a molecule from a mixture of different molecules, which makes the central theme or basis of biochemical analysis. After that, most of the reactions taking place in our body, they are reversible reactions. Still, they take place in one direction only. So how that direction is controlled? That direction is controlled by three principles. One, the principle of chemical equilibrium. Second is redox potential. And third is thermodynamics. Now let us talk about the chemical equilibrium. You know that in a reaction A plus B combine to form C plus D. In this case, the rate of reaction of formation of C plus D, V1 will be proportional to active mass of A into active mass of B, and V1 will be equal to a constant K1 active mass of A into active mass of B. Similarly, for the backward reaction also, after some time what will happen, some C and D molecules will start uniting to make again some A and B molecules. And for that, again V2 will be equal to another constant into active mass of C, active mass of D. At equilibrium, both reactions V1 will be equal to V2, so both fractions will become equal. Now what will happen? After equilibrium, how the direction of reaction will be controlled. Now, after equilibrium, as much A and B molecule will combine to form C and D, same amount of C and D molecules will again combine to form A and B. It doesn't mean that the reaction will stop. The reaction will continue, but both reactions will remain equal at that time. So how to disturb that equilibrium? If we add A or B, again some of A and B molecule will combine to form C and D. Or another way is, if some of the C or D molecules will be removed, then again some A and B molecule will combine to form C and D. Vice versa, if some C and D molecules are added, then the direction of reaction will be on this side. Or if A or B molecules are removed, then also the reaction will take place in this direction. So this way you will study later on in cellular transport of carbon dioxide and many other reactions, in which reaction is the same, but it takes place in opposite direction in different circumstances. In universe, any movement of energy follows the laws of thermodynamics. First law of thermodynamics is the law of conservation of energy, that energy can neither be created nor destroyed, although its form can be changed. For example, the electrical energy coming in our house from powerhouse, if it goes into the tube light, it changes into light. If it goes into the ceiling fan, it changes into mechanical energy. In a heater, it changes into heat energy. So whenever energy can go from one place to another, but it can neither be created nor destroyed, it is the first law. The second law is that if energy goes from one place to another, only a fraction of energy becomes available to us for performing a work. A part of the fraction of energy becomes unavailable to us, which is called entropy. So second law of thermodynamics says that free energy is equal to enthalpy minus entropy. Free energy, the energy which becomes available to us, it is also called delta E. Enthalpy is the total energy content of a system, it is called delta H. And entropy is designated by T delta S. T is the absolute temperature. So those reactions in which there is loss of entropy will take place spontaneously. For example, melting of an ice into water, it will take place automatically. But if we wish to change water into ice, there is a need of gain of entropy. It will take time, it will take effort. So what this entropy is? Entropy is a measurement of disorder or randomness of a reaction. Then you will ask, what is randomness or disorder? You can visualize it by an example. Suppose in a room, on the stool, a perfume bottle has been kept. This arrangement is ordered, or also called less random. After some time, what happens? Suppose this perfume bottle falls from the stool and all perfume molecules get distributed uniformly all over in the room. So it will be called disordered, or it can also be called more random. So the molecules have a tendency that always they try to change from an ordered arrangement into the disordered arrangement, or from a less random structure to more random. So that's why the measurement of this randomness or disorder of a reaction is called entropy. So what will happen? In our living system also, for reversible reactions at many places, for example in the reactions of glycolysis, Krebs cycle, etc., we can see that reactions from glucose to glucose 6-phosphate takes place easily, but the reverse reaction needs much more effort. So those reactions in which there is loss of entropy, that reaction will take place comfortably. Where there is a gain of entropy, that reaction will take place in a difficulty. The third concept is redox potential. You know, when any group, radical or molecule, combines with oxygen, it is called oxidation. Or where there is removal of hydrogen, it is also oxidation or dehydrogenation. Or there is a loss of electron, that is also an oxidation. If there is a gain of hydrogen, loss of oxygen, or gain of electron, it is called reduction. The capacity of different atoms and radicals to be oxidized and to be reduced depends upon the number of electrons in their outermost orbit and the electronegativity. So what will happen? You know that in periodic table, on one side we have hydrogen, sodium, potassium, lithium, rubidium, cesium, and on the other hand we have halogens, fluorine, chlorine, bromine, iodine, astatine, etc. So different elements have a different status of oxidation and reduction. For comparing this capacity, we compare it with a standard electrode potential of a standard, whether hydrogen electrode or calomel electrode. So what will happen? Direction of reaction is also controlled by a change in the redox potential. And where there is a redox potential change, the molecule will react in the forward direction, and there is a less change in redox potential, the reverse reaction will take place with a difficulty. So at many places in biochemical reactions, there is a gradation of redox potential among the different reactions of the cycle, so they control the direction of the reaction. So this is the basis of biochemistry. After that, from the next turn, we will study about different biomolecules. The first important molecule in our body is water. because it makes 70 to 75% of the total volume. Then there are carbohydrates, then there are proteins, then there are lipids and their metabolism, and there are vitamins plus mineral ions. So we will study about these molecules one by one. On the next turn, we will study about water, how the water molecule is important for living system. Thank you very much.